The title of our project is Bioinformatics Pipeline to calculate the frequency of SARS-CoV-2 variants of concern versus time and web application visualization. My name is Anabrit and I go to Mountain View High School. We worked alongside Angelina, who goes to Crystal Springs Upland School.
I'm Angela, I go to Lynbrook High School.
My name is Lucia and I go to Colegio Bolívar.
My name is Kira and I go to Lexington High School.
And we worked alongside Dr. Jepoliano Chavez.
Throughout the summer, we studied single nucleotide polymorphisms in the SARS-CoV-2 virus, which often led to the creation of new variants of concern. Through our work, we seek to understand the viral evolution of SARS-CoV-2 and its VOCs over various geographic regions using SNP identification in a three-step in silico pipeline. For further application, we also began the development of a web application for the visualization of user-inputted sets of sequences.
We looked at a total of 10 different countries highlighted in the map shown, along with viral strains from the Chinese rufous horseshoe bat and the Sunda pangolin. From the 10 countries, we analyzed data across 3 different time periods: July to December of 2020, January to February of 2021, and March to April of 2021. Choosing our data from a broad range of locations and collection dates across the globe enabled us to get a varied sample set. By looking at the data of different time ranges, we were also able to see the progression of the virus and its virulence. We gathered our data from the Global Initiative on Sharing Avian Influenza Data, or GIVSAID, and used RStudio to run code and generate the pipeline. Specifically, we ran all the sequences through SAM tools and BCF tools, which are used for variant calling and converting the original data files into BCF files. In general, the pipeline has 5 main steps: indexing done through BWA, alignment, identification of variants through a comparison with the reference file, which is a sequence collected from one of the first COVID-19 patients, extraction, and calculation of frequencies from 27 specific mutations.
When looking at a smaller subset of the master spreadsheet, certain SNPs show up within the human COVID genome, but not within that of the bat or pangolin. 23403 would likely have allowed it to make the zoonotic jump, as its position within the gene encoding the spike glycoprotein is important in determining host range. If we compare the heat map to the phylogenetic tree, we can see how the bat and pangolin form their own cluster and differ significantly from the human genome. As the values progress to March to April 2021, the frequencies 23063 and 23603, which are a part of the mutations N501Y and P601H respectively, significantly increase over 6 times its original value. The mutations are responsible for greater resistance to antibodies, which would allow it to evade the immune system. SNPs 241, 3037, 14408, and 23403 stayed relatively the same, as they play a large role in increased transmissibility of the virus when combined together.
On another note, we used a genome browser called IGV to visualize the VCF files obtained from the pipeline. In the image to the left, the gray lines represent mutations. Therefore, this allows us to see how mutations are distributed across an entire genome. We are also able to make comparisons between different time periods. In the pictures to the right, we created a chronological comparison using mutation N501Y and sample files from Brazil, from July 2020 and January 2021. The red square is located in the location of mutation N501Y. We can see that in the samples from July of 2020, none of them have that mutation. Meanwhile, in January of 2021, 3 out of the 7 files have the mutation. We use this data along with other further data to create the line chart in the bottom, which shows the frequency of this mutation since July 2020 to April 2021. As we can see, the mutation has continued to increase, and we can predict that right now it's even at a higher percentage. Also, we used a software called Paimo, which is a protein modeling software to create an analysis of the SARS-CoV-2 spike protein. In the picture to the left, you can see that we modeled the spike protein. The part signifies the receptor binding domain, while the blue part signifies the N-terminus domain. In the part of the result, we have 2 images. Image A does not include the N501Y mutation, while image B does. From these images, we can conclude that the mutation leads to an increased number of polar interactions, or hydrogen bonds, which increases the affinity between the spike protein and the ACE2 receptor in the cell. This indicates that transmissibility could potentially be increased. Also, it is important to note that this mutation is found in variants from the UK, South Africa, and Brazil.
In the meantime, we also worked on developing a web application to visualize our results. We used language R and the library Shiny to streamline the process. The basic code structure is shown on the slide. So for our app, we accepted user input of FASTA files, and we created frequency tables and phylogenetic trees. So the frequency table shows the first 10 positions in the sequence that has been aligned through multiple sequence alignment, and it shows the number of sequences that have each nucleotide or amino acid at each position. So we used sequence data from the SARS-CoV-2 genome, as well as the protein sequence from the spike protein specifically. And for a phylogenetic tree, we used the neighbor joining technique in order to increase the speed at which the web app functions. This app is still currently in development, and the data we used was mostly for demonstration purposes.
With higher frequencies of mutations, it leads to greater changes in transmissibility and virulence. The overall trend was that as time progressed and the virus spread, the genetic variation increased, leading to greater separation among the countries. One commonality that we found was that the SNPs, which had higher frequencies, tended to affect the spike proteins, and in doing so, it helps the virus to be able to bind more effectively to the ACE2 receptors on the cell itself.
In terms of future research, we have one concrete idea for the web app, that is to connect it with the data from the pipeline and create a line graph so that the frequency of variants can be monitored over time, and health policymakers will be able to use this information to better guide future actions against the pandemic.
More generally, we'd like to continue tracking the evolution of SARS-CoV-2 variants as data from around the world continues to be collected throughout this pandemic. More specifically, we'd explore various mutations and their impact on viral abilities, such as zoonosis properties. Finally, we will streamline the data analysis process for the future in preparation for possible pandemics. Thank you.